This next topic is really, really highly tested on tests, and it's really important that you understand it. It's more of, this is really a concept that really gets applied to a lot of test questions, and I can kind of tell you how it's applied. But the whole concept of having channels on a cardiac myocyte, so that's what this red box is supposed to be, is it's supposed to be a myocyte it's a cardiac myocyte. Anyway, okay, so there's a few types of channels that you need to know about. So channels and exchangers, I should say. So these black dots are exchangers. There's a exchanger where I believe it's 3 sodiums for 1 calcium. I don't know why there's 2 on there. I was probably testing you. And then there's another one, and this is everywhere. Every cell has a sodium-potassium ATPase. You can see here that there's 3 sodiums going out for 2 potassiums going in, and it's kind of hard to see here, but you can see that the shell of hydration around a sodium is actually larger than that around a potassium. And so that's a big theme in physiology, is that sodium and volume are almost synonymous. Water follows sodium more than it follows potassium. And if you look at a periodic table, which you probably haven't in a while, you'll notice that that sodium is actually in the same row as potassium, but sodium is above it, meaning that it's smaller. Okay, so why does sodium have a larger shell of hydration? Well, it's because they both have a positive 1 charge, but sodium's smaller, so its charge density is much, much more than, than potassium. So therefore, water is more attracted to it, and when you have things like the kidney and stuff like that, which you'll talk about some other time, water follows the sodium. So anyway, but the sodium-potassium pump is a huge thing in physiology. There's a lecture that you'll have in your second year of medical school where somebody's going to make the point that life is based on the sodium-potassium pump, and that the ability to store energy in gradients is really how everything works. Anyway, so with this heart cell, you have the sodium-potassium pump, and then so sodium goes out, potassium comes in. Again, that's kind of a big topic in physiology and in the body, is that our cells spend a lot of energy keeping sodium out and potassium in. And blood, let me just give you a few numbers here. Blood sodium is going to be 135 to 145 milliequivalents per liter, whereas blood potassium is going to be 3.5 to 5.2. So that's inside the blood. So that's going to be the opposite of inside the cell. So the cell is going to be the exact opposite of that. You're going to have high potassium, and you're going to have low sodium. Okay, so that's enough of an introduction. But you can see how these different things work. So, okay, let's talk about a few applications. Okay, so again, you can just kind of see that to get calcium out, you have to bring 3 sodium in, and then those 3 sodium will come over here, and they'll be exchanged for potassium. So what happens is there's a medication that's referred to as cardiac glycoside. Cardiac glycoside is actually the class, and there's really only one useful medication in that class, and it's called digoxin. But I believe that a lot of the ways in physiology it's tested is they'll say cardiac glycoside. Okay, so what cardiac glycoside does is it interferes with the sodium-potassium pump, and specifically it interferes with the potassium channel right here with that red X. And so what happens is if you shut down this pump, then these sodium are not being transferred out, and these potassium are not being transferred in. And so when you shut down that pump, then that means that that's going to affect here, because we're going to have a buildup of sodium in here. And so these 3 sodium that want to come in are going to have a harder time coming in, which means that this calcium that's going to go out is going to have a harder time going out. So you're going to be kind of effectively shutting down that exchanger also. And so what's going to happen is inside the cell, you're going to have an increase in calcium. Okay? And calcium is synonymous with contractility. The more calcium, the more contractility, because the calcium is what allows the actin and myosin to interact. So the more calcium there, the harder that contraction is going to be. And so that's a really heavily tested concept, is giving this medication causes contractility to increase. So somebody with heart failure, you can give them this medication, and the analogy that I've heard, and you'll probably hear in your second year of medical school, is you can either do one thing to a donkey, which I'm not going to mention, or you can whip the sick donkey, and giving cardiac glycoside or digoxin is an example of whipping a sick donkey. You're making that failed heart work harder, and you're keeping calcium inside the cell, and so the cardiac myocytes that are available and remaining are going to work harder to make the heart work. Okay, and so a few other things about this. You've hopefully remember beta 1 agonists. Beta 1 being a receptor, it's an adrenergic receptor. It's a sympathetic adrenergic receptor. And do you remember what type of G protein it's associated with? It's going to be a G stimulatory, and you can tell that from right here because we have an increase in cyclic AMP, and that increase in cyclic AMP is going to signal for more calcium to enter the cell. So when we get chased by that bear, our sympathetics are going like crazy. What's happening is the epinephrine or the norepinephrine that is stimulating the beta 1 receptor on the heart is increasing the calcium inside the heart, which is making the heart rate go faster. Or actually, it's not technically making the heart rate go faster, it's actually making the heart squeeze harder. It'd be the SA node and the AV node that are making the heart go faster. Okay, so then parasympathetics, they want to help out too. So they use acetylcholine as their transmitter, and they transmit to a receptor. And if you remember, that'd be an M2 receptor. And so what that M2 receptor does is it's a G protein also, and it's going to be a GI protein, G inhibitory protein, and it's going to do the exact opposite. So we have increases in cyclic AMP from the beta 1 receptor, and we have decreases in the cyclic AMP from the M2 receptor. So that's how sympathetics and parasympathetics, they oppose each other. Side question, does anybody remember which one dominates how the heart works? Kind of goes back a test or so, I think. It's actually the parasympathetics dominate. So kind of counterintuitive, but they do. So, and the way the parasympathetics works is they increase the potassium permeability. So if you remember, the potassium potential, and I don't want to get back into cell too much, but the potassium potential, I want to say it's like -90 to a -100. And so the more permeable you make the cell to potassium, the closer you're going to get to the potassium potential, which is -90 to -100. And so that cell membrane is going to become hyperpolarized, and it's become less likely that an action potential is going to take place, which will then lead to the coupling of the muscle contraction and the electrical activity, which don't want to get into that here, but that's how heart rate is decreased, and also the contractility is decreased through parasympathetics. So anyway, the big thing here is just understanding what's going on. This is something that you don't want to memorize. This is something you want to understand. And if you understand it, you'll get the test question right every time. And I recommend drawing out a little drawing to try to to try and make things make sense for yourself, and to also do that on test questions. And hopefully that helps you.